If we treat human aging as a solvable evolutionary problem rather than inevitable mechanical wear and tear, the path to clinical control becomes clear. By shifting focus to evolutionary genomics, we can effectively manage the aging process within 50 years. This strategy follows the same historical blueprint used in the 20th century to map and conquer infectious diseases that once decimated global populations. We now have the evolutionary framework to neutralize aging with that level of systematic precision. Before the 1860s, doctors blamed cholera and plague on miasma, bad air. Today, progress on aging is stalled by a similar misconception: the idea that the body simply breaks down like a machine. In reality, aging is a complex, polygenic network failure. It occurs because the force of natural selection drops as we get older, leading to a loss of genetic adaptation in late adulthood. This timeline tracks the progression from the discovery of germ theory to the widespread use of vaccines. Applying an evolutionary framework to aging follows these same milestones, moving from public health reform to targeted clinical action, and eventually total management. Viewing aging as a predictable evolutionary decline is the essential foundation for all modern anti-aging medicine. Phase 1 focuses on the first 10 years. We mitigate age-related decline by adopting an ancestral paleo diet and walking 3 to 5 miles daily, beginning after the age of 30. This graph plots biological phenotype against adult age. The top line represents the optimal state for a new environment, while the curve below tracks our evolutionary response. Because natural selection falls with age, older individuals lack the genetic adaptation to survive modern, sedentary agricultural environments. The gap widens as we get older. Industrial diets and inactivity harm older populations at a much higher rate than their younger counterparts, who have had more time to adapt. This lifestyle alignment is an essential triage step. It buys older generations the time required for advanced therapeutics to arrive. Phase 2 spans years 10 to 30. Since aging involves hundreds of genes, clinical medicine must attack the symptoms individually rather than searching for a single master switch. We will deploy autologous stem cells for tissue replacement and use real-time monitoring to disrupt physiological death spirals before they become fatal. This stacked bar chart shows how treating these individual failures pushes the mortality plateau further to the right, adding decades of functional life. By preventing acute systemic failure, these targeted interventions provide the clinical bridge to complete biological control. Phase 3, years 30 to 50, moves to the genomic level. Managing aging is a massive computational network challenge involving hundreds of interacting genes. We can solve this network problem because humans share over 80% genetic orthology with specific model organisms like the fruit fly. This data network graph shows the transition from a model organism to the complex human genome. By isolating these overlapping nodes of orthology, we can develop pharmaceuticals that retune human genetics for late-life health. This genomic retuning addresses the root of biological aging at the cellular level. Physiological deterioration is not an absolute rule. In many species, and historically in humans, aging naturally plateaus into a stable state. Our 50-year roadmap relies on 3 pillars: surviving the near term through lifestyle changes, buying time with tissue repair, and eventually solving the genomic code. The biology of defeating aging is now established. Success is a matter of sustained execution and focused resources.